Thank you so much for the kind introduction, and I'm really, it's my pleasure and I'm honored to be here and speak along with my esteemed colleagues with the great panel that we had this morning. This is a milestone year for the US with a few biosimilars, especially the monoclonal antibody pertinent to the rheumatology community, haven't been approved by the agency, and also increasing knowledge and potentially one close to launch and being used in the community in near future. We are all sitting here talking about different issues around biosimilars. And obviously, the regulators and industry had this for a period of time. And it's the rheumatology community and the other physician community. With the newness of all of this, we are struggling to understand the differences from innovator, the pathway that we have been so familiar for a period of time. So I must say that I here will present Amgen's perspective, and Amgen is not only biosimilar developer, it's an equal innovator developer under the same umbrella. So Amgen is equally committed to bringing new molecules, innovator molecules for the patients, as is committed to bring the biosimilars for the patients as well. So this is my conflict of interest. I am an employee and stockholder of Amgen. And what I'm going to do is actually on the fly, because we had such great presentations since morning. And a lot of, as Mark very well said, a lot of information is limited information, limited data set. We are pondering on the same issues. So I'm going to make my talk, last but not the least, more interesting before you hear from Stephen from cost effectiveness. So I'm going to hit certain points, and I do have some examples which can help everybody understand things better from Amgen's perspective, as well as from biosimilars development. So you go with a positive approach towards biosimilars, and also some of the key points that have been pondering on everybody's mind that are addressed. So I'll talk about functional similarity. I'll talk about the appropriateness of the clinical trials. I'll talk slightly about the single transition and the interchangeability, which has been all under discussion, and I'll be happy to answer any questions. Now, biosimilar manufacturing comes from, prefaced by biologic manufacturing, and we know that biologics have been so important in our particular subspecialty. And I had the privilege to be trained in the era when biologics were available with my trainee, Dr. DeHoratius here. So, I have not had the opportunity to really see a lot of patients on wheelchair, on IV goals, some of the things a lot of audience have seen, but I do have a lot of respect for that. So biologics have changed the way patients are being treated, but we all understand that the manufacturing of biologic is very, very complex. And here, this slide particularly depicts just 4 big buckets of how the manufacturing occurs. It's a tedious process, it's very complicated, and it starts from taking the molecules before even animals. It starts from that standpoint, and it continues to keep going iteratively till the time of approval. Following approval, and it's a part of the approval package, so it's very tightly regulated. Following approval, any of the changes which the manufacturer will make is already based on the knowledge that they've developed doing this over and over again for maybe a decade or 6, 7, 8 years at period of time. So that exercise is called comparability, which you've heard is manufacturing change. What I'm trying to differentiate is that from biosimilarity, and this process is very tightly regulated, as a lot of previous speakers talked about. So this is called comparability, and there is a comparability assessment that occurs. Dr. Strand's slide referred to that in the morning. It depends on the risk of the change, and it is very tightly controlled. So you might see some information, you might not see information at all. But I think you have to take it for the regulators' world that it is very tightly controlled process, and it's not the same as creating a biosimilar. Biosimilar manufacturing, and you've seen some version of this slide before, is very different. The biosimilar manufacturer starts really from the literature that's available for the reference product, which is the innovator product they are developing a biosimilar to. There are few unknowns. There are a lot of few knowns, a lot of unknowns. And then they start to do their own experiments. So we will take the literature available, we will procure the reference product over a period of time, we will study it, do multiple experiments to really understand and outline it better before we actually start to develop it. Once you've underlined and outlined it, that's when you characterize, you call it characterization of the reference product. And each product has its own special characteristics, which are called the CQAs, which make it important to impact the clinical efficacy. It's going to be different from one molecule to another. Those are very important to outline, and they get prioritized in discussions with the agencies. That's when an experienced biosimilar manufacturer comes in, and they will use their experience, expertise to develop a unique cell line. So, we know by virtue of the way they are created, there are going to be differences, and they're accepted and expected, minor differences as the regulation says itself. And each biosimilar will slightly differ from another biosimilar as well. So, once the CQAs are identified, the unique cell line, the unique manufacturing process that each biosimilar sponsor will take to create their own biosimilar, they will have multiple biosimilar candidates, which could be clones, and they do multiple of these iterative exercises, which are called similarity exercises, to say which one is the closest of the clone to the reference product. And that happens in discussions with the agencies as the clones are selected. And as Dr. Woodcock said, there are multiple touch points to ensure that a highly similar biosimilars move forwards into the patients. So then the similarity assessment continues on as the biosimilar candidate moves forward into the preclinical animal studies, if needed be, phase 1, phase 3 studies. The similarity process continues to keep going on till the time of approval, and all those make substantial data, which makes the bottom of the pyramid. If the pyramid, you remember from this morning, and I will not show you the pyramid per se. So looking at the critical quality attributes, there are many, many different ways of looking at it. You identify them, you prioritize them, and based on the state-of-art methods that are available, the assays that are available, the biosimilar manufacturer will actually use all of them to look at them orthogonally, which means if this is a one critical quality attribute, which could be a structure or a function, something related to stability, there are multiple tests done to look at it many different ways to ascertain that there is similarity of these key critical attributes. And it could be on an average, there are over 100 attributes which are looked at across 40 or more assays. Now, it is the heterogeneous structure of the biologic itself, that as well as the production process of it, which will lead some residual uncertainty. And at any given time, you have to understand what is really critical to the molecule, to its mechanism of action and the clinical activities, and what is not, and how can we best assess that to avoid the impact. That's where the design consideration involves the mechanism of action, and these are called the biological or the functional activities. So the structure of the molecule itself, the IgG1, there are plenty, the different parts of the molecule have plenty of functional activities, and that's why I'm emphasizing that looking at these functional activities are equally important in assessment of the biosimilarity. For example, if you will, for monoclonal antibodies with anti-TNFs, some of the key functional activities are actually defined there, which is target affinity, binding affinity, target neutralization, looking at apoptosis, effector function activities such as CDC and ADCC, and FC binding. And now I will show you the example of ABP 501, which is Amgen recently approved, where it was evaluated for potency, FcR and binding, CDC and ADCC activities to show similar biological functional activities. So Understanding the structure is important, understanding that there might be slight differences in the structure that could exist by the inherent nature of the biologics and creation of biosimilars. And I will show you some examples of how the structural differences, which are small differences, do not impact the functional activities, which is important and key to the mechanism of action. And these have all been our own experience while creating many different biosimilars within Amgen. So this is an example one where we looked at glycosylation, and as we see, on the upper right corner, we have the reference product in the red, and you can see the biosimilar in the blue. And you can see the quality range, I think, has been mentioned, is actually defined by each biosimilar sponsor per se. And this quality range is defined by, procurement and testing of all the different reference product lot that the biosimilar manufacturer procures and creates a range for themselves. And then they test themselves head-to-head with the reference product. So even though we see there are a few which are outside the predefined quality range by a few percent, when we evaluated this in a functional activities of relative CDC, it actually did not matter. So this gives assurance that the functional activity, which is key to the mechanism of action of the molecule, is sustained, and it's in the predefined range. Next is another example of another biosimilar where we're looking at the total afucosylation. And total afucosylation at the top right upper corner is a product of the high mannose, which we've heard in the talks in the morning can impact PK as well as the afucosylation. So all of these are contributory to the relative ADCC activities. Now you can see slight differences up and down in terms of the reference and the biosimilar per se. But when we looked at the ADCC activity, it was spot on for the biosimilar within the reference range for the reference product. So as the biosimilar manufacturer is creating their own process, they have, if you will, these models, experimental models, which they can do slight tweaking to tighten things up to make sure that the function, which is key to the mechanism of action, is actually maintained. So hopefully with this, I'll give you an extreme example of actually the host cell expression change. So I think most of us are familiar with a CHO cell line versus sp2/0 cell line. So I'm giving you an example which has been published, presented at EULAR by Dr. Bourne et al. At Amgen. And they were looking, by creating biosimilars, looking at a CHO versus a CHO, similar host cell expression lines. I must say here that each cell line will be proprietary to each manufacturer. So they can come from the single parent umbrella of CHO, and there will still be differences between what one sponsor uses versus another. So what they tested here was looking at the two similar CHO cell lines of the reference and the biosimilar candidate, and took it through a process. And as you can see maybe I'll walk you through it. This is the antibody concentration, and on the y-axis is the relative ADCC activities. So the reference is in blue, and you can see as the antibody concentration increases, the ADCC activity increases for all of them. However, when the biosimilar was created with a process 1, and then tweaked to 1A and 1B, or process 2, even though with the same similar host cell expression system, I must say, that we did not achieve similar ADCC results. So that's one example. Eventually, the process was completely overhauled to come up with new results. But this is an understanding. This is something you might see in future, so it's good for you to start to understand. So the second is now taking two different expression systems, an sp2/0 system and the CHO cell system. We understand that there are differences inherently from the parent cell lines that are going to lead to some structural differences. However, with careful engineering of these molecules, the functional activities of these molecules are spot on to each other. So it is very important to match the functional activity for the assessment of biosimilarity. So this is the first chapter, if you will, from my discussion on the structure and the function of this. Then I'll quickly go into the clinical consideration. I think a lot of it, Vibeke has already covered in the morning. I will emphasize a few points. One is, I've seen a lot of discussion, confusion around what is an equivalent study versus what is a non-superiority. So an equivalent studies are intended to demonstrate clinical equivalence in the totality of evidence package for biosimilarity. So you have to show that you have a similar effect to the comparator or the reference product. And the null hypothesis, if you will, with the predefined margins actually has to show that the biosimilar is non-inferior. It's a two-sided test. Non-inferior and non-superior both at the same time. And it's a very different concept from a non-inferiority study or from a superiority study, which most of us are very familiar with based on our knowledge of the new agents. So this is a different testing concept, if you will. And then I will depict it with a picture helping you understand that this is what equivalent means, is you are non-superior and non-inferior both at the same time. Other considerations now, this is the exercise, if you will, is not to show the clinical efficacy de novo. The exercise here is putting the biosimilar and the reference product in many different experimental conditions, and the end result you're showing is they're similar to each other. And that's the totality of evidence package that you're actually, as a sponsor, collecting. And at the end, which is the clinical trials, which are, rough in terms of assessment, the assessment, if you will, compared to the very sensitive and robust analytical testing. When you get to the clinical trials, the exercise is, this is a new sensitive experimental condition involving patients. So you have to pick up the sensitive patients, the sensitive endpoints, sensitive indications. The duration could be very different. And the treatment regimen are going to be very different compared to what has been done for the innovator, because the answer that is, the question that we're answering from this experiment is just inherently different. It is showing the last step of the biosimilarity assessment, versus innovator has a higher burden of showing treatment effect, safety, as well as efficacy against a placebo. So therefore, the indications sometimes might not be the most sensitive indications, or they have to be the most sensitive indications, but sometimes they might not be the commonest ones, which are actually, the product is actually being used. So you'll see a difference right there. The comorbidities, the con-meds could be different, and really this particular slide brings in perspective both for inflam as well as oncology standpoint. As I move to the next slide is really referring to what you will see in most of the anti-TNF therapies is use of either rheumatoid arthritis or psoriasis as the indications which are being studied. And I think it has been well outlined in the morning. With psoriasis, there is a monotherapy, and there is no, the background now is, if you will, is actually very low in terms of con-meds, the disease conditions. The effect size is really high. When the effect size, the treatment effect is high, that helps you distinguish if there were any differences between both the reference and the innovator, that makes it the most appropriate population to study. Then the disease severity could be moderate to severe, as the treatment regimen could be with methotrexate or without methotrexate. The endpoints might not necessarily be the endpoints that you're used to familiar with, and this is actually even more pertinent in oncology. However, a PASI % improvement, which is a continuous variable, which brings in all the variability from the beginning to end, is probably more appropriate in the situation of biosimilarity compared to a PASI 75, which is a binary cut of it. And similarly, both ACR and DAS28 C-reactive protein, they are appropriate endpoints. And it's really going to depend on where the justification of margin will come on, how much data is actually available to actually create the margin around it. And this is really, in discussion with the regulators too. Then you will also see study designs which are different. It could be just 2 parallel arms, no switch, one switch or one transition, or multi-transition. So with that, in the last few minutes, I will walk you through the single transition, and as we have done it for ABP 501. So biosimilar sponsors may study the single interchange or single transition, from the reference product. And this was particularly studied with ABP 501 in the psoriasis study, in the moderate to severe plaque psoriasis, primary endpoint at week 16. And thereafter, the study continued. The ABP 501 group at the top row continued as such. The adalimumab group got divvied up into either continuing on adalimumab or ABP 501. So this is a situation of single transition, and as I think it's been very well defined from the morning sessions, that the single transition brings in information about, the single transition brings in information about safety, immunogenicity, and long-term efficacy, if you will. So those were all studied for ABP 501, and I'll walk you through the results. So this is looking at the PASI % improvement for the single transition arm, which is in yellow for adalimumab to ABP 501. And this is a continuous variable looked out as a primary endpoint throughout the study. And this is looking at the binary cuts, the various binary cuts of the PASI 50, 75, 90, and 100 at week 50 for all the three groups, including the single transition group. So key is also looking at immunogenicity of this. So this is the week 4 and week 16, and after that the groups split. And then this is looking at week 16 to 52 with the yellow for the single transition group. And we also did more analysis because in immunogenicity in monoclonal antibodies, especially the ones that we use for rheumatology, the immunogenicity rates being very high can impact what you can see after week 16 is potentially impacted by what has already developed at or before week 16. So we looked at the clean group. We took the patients who were ADA negative at or before week 16, and we looked at the binding antibody development for those patients after week 16 to really clearly understand the immunogenicity. And we see through the, from that point onwards till the end of the study, really the immunogenicity was similar for the switch or the single transition group as well. And the safety was comparable as well. So with that, I will move on to the discussion of the day. And I'm going to try and make it a little different, bringing our own perspective on interchangeability. So we know from all the discussions since morning, biosimilarity is being highly similar and notwithstanding minor differences in the clinically inactive components, and no clinically meaningful differences in terms of the purity, potency, and the safety of the molecule. And we also have learned that interchangeable is actually a higher standard. You need to first be a biosimilar before you can actually think of being interchangeable. Interchangeability will include, number one, being a biosimilar. Number two, showing the expectation of the same result in any given patient. And number three, for the product that is administered more than one, there's no additional risk to safety or diminished efficacy as a result of switching. So this is what it looks like from Dorner and Kay's publication. So really, when we think about this, and I'm really going to end here, just trying to give some food for thoughts of what interchangeability should look like, or Amgen's perspective on it, it would be essentially trying to study a worst-case scenario where you provide an exposure to a patient such that you boost their immune response. So if we had to look for that, what would we think of the endpoints? In my mind, the clinical efficacy actually would not be the most appropriate endpoint in such assessment, because there is a hangover effect, if you will, for clinical efficacy. We know that withdrawal and relapse of clinical efficacy just with the anti-TNFs takes a period of time. So in trying to study such such a design of clinical trial, this would not be the most appropriate. And as it is, clinical efficacy would be, or the lack of clinical efficacy would be, in fact, the result of, probably development of ADA, because we are boosting the immune system such that anti-drug antibodies are developing differentially, and that could impact the kinetics of it. So I would leave everybody with the food for thought that the sensitive assessments in interchangeability potentially would be pharmacokinetics and immunogenicity. And with that, I will thank everybody for their time.